Summative test for Chapter 5. Analysis. So number 1, define oxidation, reduction, and differentiate the following terms. So oxidation reduction, it is the involve the transfer of electrons between electron donor and the electron acceptor. So it must occur simultaneously. If an atom loses their electrons, another atom must take them. So for letter A, the aerobic and anaerobic respiration. So aerobic respiration requires oxygen to proceed, while anaerobic respiration does not, or the absence of oxygen. So for letter B is respiration and fermentation. Respiration is a set of chemical reactions involved in the production of energy by completely oxidizing food and oxygen required, while in fermentation is chemical breakdown of organic substances by microorganisms, typically of effervescence, and the heat and oxygen not required. So for the letter C is the cyclic and non-cyclic photophosphorylation. So cyclic is synthesize only ATP. In this process, electrons move in a closed circle, ATP and NADPH2. In this process, photolysis of water takes place and oxygen is liberated. So for number 2, all of the energy producing biochemical reactions that occur in cells, such as photophosphorylation and glycolysis, are called the oxidation reactions. So for number 3, fill in the following table with the carbon source and energy source of each type of organism. So the first organism is the photoautotrope. So the carbon source is carbon dioxide and the energy source is light. For the next organism is photoheterotrope. Their carbon source is organic molecules and the energy source is the light. Next one is chemoautotrope. So the carbon source is carbon dioxide and the energy source is inorganic molecules. So for the last organism is the chemoheterotrope. The carbon source is organic molecules and the energy source is organic molecules. So for critical thinking, question number 1 is that explain why even under the ideal conditions, a streptococcus grows slowly. It is because what they use, which is an aerobic respiration, does produce a little amount of ATP. So for question number 2, compare and contrast carbohydrate catabolism and energy production in the following bacteria. So the first bacteria is Pseudomonas, an aerobic chemoheterotrope. Most of cell's energy is produced from the oxidation of carbohydrate. It is usually a glucose. Between this the common source of carbohydrate. Energy is produced from complete oxidation of one glucose molecule using aerobic respiration. So the next one is the spirulina, an oxygenic photoautotrope. As a source of energy, they use carbon dioxide as a roll of carbon. They use water to reduce carbon dioxide and uses light-dependent reactions to absorb light to form oxygen, ATP, and NADPH. So for question number 3 is that how much ATP could be obtained from the complete oxidation of one molecule of glucose? From one molecule of butter fat containing 1 glycerol and 3 12 carbon chains. So we have here a 38 ATP could be obtained from one molecule of glucose that was completely oxidized, and 100 ATP could be obtained from one molecule of butter fat containing 1 glycerol and 3 12 carbon chains. So for question number 4 is that the chemoautotrope thiobacillus can obtain energy from the oxidation of arsenic. How does this reaction provide energy? How can this bacterium be put to use by humans? So, due to the formation of proton radiant during the arsenic oxidation, the energy that bacteria obtain from the arsenic. So, the arsenate that is formed is poisonous but can be used chemically. So for question number 5 is that Haemophilus influenza requires hemine, the X factor to synthesize cytochromes, and NAD to the positive ions. So V factor from other cells. For what does it use these two growth factors? And what disease does Haemophilus influenza cause? So it may cause a variety disease and such a meningitis, meningitis, inflammation of the waving coverings of the spinal column and the brain. So bloodstream and the infections, pneumonia, arthritis, and infections of other parts of the body due to the widespread of uh, of vaccine in children uh, and common causes of bacterial infection. So for the last question is number 6. Um, this bacteria enzyme streptokinase is used to digest fibrin or blood clots in patients with atherosclerosis. Why does an injection of streptokinase cause a streptococcal infection? How do we know the streptokinase will digest fibrin only and not good tissue? So, the indirectly break down blood cells via the um, activation of the another enzyme known as the plasminogen, which is ultimately cleaves into plasmin, which is the action taken in a breakdown dots. Streptokinase is only an enzyme and not a weakened pathogen such as vaccines. Streptococcus won't invade the toys. Enzyme don't cause disease. Virus and infection cause disease. Summative test for chapter 6, the analysis. Number 1, describe binary fission. Binary fission is a process which asexual reproduction occurs in organisms through the split of parent organism into two and the regeneration of the two pieces. It requires only on the organisms of the single parents and occurs in both prokaryotes and eukaryotes. So for question number 2, macronutrients needed in relatively large amount are often listed as CHONPS. What does each of these letters indicate and why are they needed by the cell? So for the first letter is letter C, which is the carbon. its synthesis of molecules that make up living cell. So for the next one letter is the H, which is hydrogen. It's the source of electrons and component of organic molecule. For the next one is letter N, which is the nitrogen, that amino acid component. The next one is the O, which is oxygen, is organic molecules, component, electrons acceptor, and aerobes. The next one is the P, which is phosphorus, in phospholipids and nucleic acids. Next, and the last one is letter S, which is the sulfur in some of amino acids. So for the question number 3 is define and explain the importance of each of the following. Number 1 is the catalase. Catalase, it is the antioxidant enzyme which is produced naturally in all living organisms. It also scavenges reaction oxygen species within the cell. It degrades hydrogen peroxide into oxygen gas and water molecule. Catalase is constantly in battle against the effect of free radicals to the body. For the next one is the hydrogen peroxide. It is a mild antiseptic use on the skin to prevent infection of minor cuts, burns, and scrapes. They use them as a disinfectant for wounds and even on mouth prints and to remove bad odor. The next one is the peroxidase. It is a common enzyme that is important in decomposing the peroxide compounds. It also important treatments of industrial wastewater to convert toxic substances into less harmful substances. And for the next one is the superoxide radical. So it is produced by the one electron reduction of molecular oxygen. It acts as a signaling molecule regulating numerous biological processes, including apoptosis, aging, and senescence. And for the last one is the superoxide dismutase. So this is a metal containing antioxidant enzyme that reduces harmful free radicals of oxygen formed during a normal metabolic cell process to oxygen and hydrogen peroxide. And so for the question number 4 is that 7 methods of measuring microbial growth were explained in this chapter. So categorize each as either a direct or an indirect method. So it is a direct method are those in which the microorganisms are seen and counted. So it is direct microscopic count, plate count, and the filtration, and the most probable is the most probable number. So for question number 5 is that by deep freezing, bacteria can be stored without harm for extended periods. Why do refrigeration and freezing preserves food? So with decreasing temperature, the growth rate of the bacteria slows down. Mesophilic bacteria will grow slowly and refrigeration temperatures and will remain dominant in a freezer. The bacteria will not spoil and food quickly in a refrigeration. So for the question number 6, a pastry chef accidentally inoculated a cream pie with 6 aureus cells. If S aureus has a generation time of 60 minutes, how many cells would be in the cream pie after the 7 hours? So, the number of cells times the 2n generations is equal to the total number of cells, just like 6 times 7, 6 times 2 to the to the 7 power is equal to the raise of 7 is equal to 768. And so for the question number 7, nitrogen and phosphorus added to beaches following an oil encourage the growth of natural oil degrading bacteria. Explain why the bacteria do not grow if nitrogen and phosphorus are not added. So, nitrogen phosphate, nitrogen phosphate are very essential for making phospholipids, nucleic acids, proteins, and the ATP. However, they are not usually usable in a large quantities. Petroleum can meet the carbon and the energy requirements for an oil degrading bacterium. And so for the last question is that um, differentiate complex and chemically defined media. So chemically defined media are a culture media that contain the exact chemical composition is known, whereas complex media are culture media that contain an unknown chemical composition. For critical thinking, Clostridium and Streptococcus are both catalyst negative. Yes, it is. So, Streptococcus grows by fermentation. Why is Clostridium killed by oxygen whereas Streptococcus is not? So, Streptococcus is able to live in anaerobic conditions while Clostridium is not for the reason that it possesses superoxide dismutase or an equivalent system that neutralizes the toxic forms of the oxygen. So for the next question is, most laboratory media contain a fermentable carbohydrate and peptone because the majority of bacteria require carbon, nitrogen, and energy sources in these forms. How are these three needs met by glucose minimal salts medium? So, the media is often used to define if a particular microbial species is a heterotroph, namely an organism that does not have any nutritional requirements beyond core sources of nitrogen and carbon. However, minimal media contains the essential for bacterial species to grow. So, for question 4, flask A contains yeast cells and glucose minimal salts broth incubated at 30 degrees Celsius with aeration. Plus, flask B contains yeast cells in glucose minimal salt broth incubated at 30 degrees Celsius in an anaerobic jar. These yeasts are facultative anaerobes. So for letter A is, which culture produce more ATP? So plus A, if the yeast is a facultative anaerobe, it means that it refers aerobic conditions but can also live anaerobically. So it has oxygen. So the cells will be able to do aerobic cellular respiration, which produces much more ATP than the fermentation process that is conducted in anaerobic conditions. So, letter B is which culture produce more alcohol? The flask B. Alcohol is produced under anaerobic conditions when the yeast is forced to undergo fermentation. So for letter C is which culture have a shorter generation time? The flask A, because the yeast prefer to have oxygen, so they will produce more quickly in aerobic conditions. So for letter D, which culture have greater greater cell mass? So it is the flask A, because the yeast prefer to have oxygen, so they reproduce fast and have the greatest mass in aerobic conditions. So for letter E, which culture have the higher absorbance? So the higher absorbance is the flask A, which is absorbance means the amount of light absorbed when light is passes through the culture flask. It is directly proportional to the cell mass inside the cell mass inside the flask. So since the flask A will grow faster, the cell mass will be higher and the absorbance will also be higher.